Hello, everyone. My name is Dr. Iftikhar Ali, and I'm a microbiologist by profession as well as by education. And today I'm going to give you a brief introduction to what else but microbiology. And this is going to be a short presentation. It's not going to be a very long one. So we are going to just deal with the basics of microbiology, definition, what is microbiology, what is it composed of, and then the different branches of microbiology and some detail about those branches. And I hope you enjoy this. So let's start off. Firstly, what is the definition of microbiology? So microbiology is the study of microscopic organisms. No big surprises there. And what do you mean by microscopic organisms? Microscopic organisms means that organisms which cannot be visualized with the naked eye, they can only be seen through a microscope. And examples of these organisms are bacteria, fungi, viruses, and also some other organisms like amoeba, malarial parasites, and even uh, worms such as tapeworm and roundworms, which, of course, they are visible to the naked eye, but they do come under the purview of microbiology. Okay, so microbiology deals with their behavior, their evolution, ecology, and by ecology, what we mean is what is the best environment for them to exist and multiply. Some of them are saprophytic, which means they are better suited for the soil, and some of them are better suited for human beings. So they are multiplying more in the human beings. And then their biochemistry, their physiology, and finally, the most important of all, the pathology of the diseases that they cause, because that is the reason why we are actually studying microbiology, right? So let's proceed on to the history of microbiology. It all started with the invention of the microscope, of course. And as we have all learned in our high school, that Anton von Leeuwenhoek was a scientist who had discovered the microscope, and he was the first one to visualize bacteria in water. And then he drew pictures of those bacteria, and they were actually published in the London Journal during those times. And he had also described some unicellular organisms, which we know now as protozoa, and he had also described some unicellular fungi, which we now know as yeast. And although the microscope was discovered and the microbes were visualized, the bacteria were seen, and they were even drawn, but still the theory of spontaneous generation of microorganisms existed even until the 19th century. Now, what was this theory? What was the spontaneous generation theory? This was the belief that living things arose spontaneously from a combination of ingredients. They did not necessarily have to have parents. They just came out of nowhere. So this was the theory of spontaneous generation. And for example, people believe that mice, which are actual animals, they arose from soiled cloth. Whenever soiled cloth was combined with wheat and then it was left out to sit, then mice can come out of that. So now this all seems very exaggerated. I mean, we may also say that silly, but that was the belief that was held even until the 19th century, and that's we are talking about just 200 years back. So this was the theory, and there were some scientists who tried to disprove it, like Francisco Redi, but this belief continued to exist until the 19th century. So who was the person who disproved this theory? It was without a doubt Louis Pasteur, which we know him for the pasteurization of milk, and the first person to discover vaccines. And he was the first person who disproved this theory by filtering out air using cotton wool. He actually isolated the microorganisms. And then he also showed that if suppose water was boiled in a swan necked flask and then kept without the air reaching the water, then it can be left sterile without any organisms growing in it. So this was what was proven by Louis Pasteur. And then, of course, the discovery of the compound microscope and the development of staining techniques, that was a big step in the type of studies that was done on bacteria and that we know now. So this was basically to visualize the organisms and basically to distinguish the different types of bacteria. And this was all done because of the discovery of the compound microscope and the discovery of staining techniques. And lastly, most importantly, people actually began to realize that microorganisms cause disease. So, this link between these organisms that were being visualized and disease, that was actually established, and that was the biggest step forward in microbiology. So, what are the branches of microbiology now? So, microbiology consists of five main branches. They are bacteriology, virology, immunology, mycology, and parasitology. So, bacteriology, as the name suggests very clearly, is the study of bacteria, their evolution, their multiplication, and then the diseases that they cause. Virology is the study of viruses, their evolution, multiplication, and then the diseases that they cause. Immunology is the study of the immune system. So this is the system which, if suppose the organisms are causing disease, then how is our body responding to those infections or that invasion by the organisms? So this is basically what is composed of the immune system, and the study of the immune system is called the immunology. And then mycology is the study of fungi and such as yeast and molds. So these are some fungi that we know now. And then parasitology is the study of parasites, and under the parasites come the amoebae, the malarial parasites, and then as we said in the beginning, some organisms which are actually can be seen with the naked eye, like tapeworm and roundworm, also come under parasitology. So that is as far as the branches of microbiology are concerned. Then coming to start of this branches of this of microbiology, the first one is bacteriology. So what is bacteriology? So this is the branch of microbiology which deals with the identification, study, and cultivation of bacteria. Okay. So what are bacteria? Bacteria are microscopic organisms. They are firstly unicellular, that is, they don't have any distinctive nuclei. They reproduce by fission or by forming spores, and they can be found everywhere. That is, they are cosmopolitan in existence, and they can exist either freely, that is, they are called free living, or they can exist dependent on other organisms, in which case they are called parasitic, and these are the ones which actually cause the diseases. So, what is the morphology? Bacteria are prokaryotes as opposed to eukaryotes. We are all eukaryotes, the fungi are eukaryotes, but bacteria are prokaryotes, which means that they lack nuclei. They don't have the regular organelles that we see in human cells or in fungal cells, like the mitochondria, the endoplasmic reticulum, the Golgi apparatus. All these things are lacking in bacteria, and hence they are coming under the category of prokaryotes. Okay, so under the eukaryotes, as I said, we have the algae, fungi, and we ourselves, animals and plant cells. And then the size, bacterial cells range from zero point 0.2 to 3 micrometers in size. And then if you compare this with RBCs, which are 8 micrometers in diameter. So just to compare, they are much smaller than a regular red blood cell. And then what are the major classes? The major classes are the gram positive, the gram negative, and the acid fast. So what do you mean by gram positive and gram negative? So these are stains which was developed by a scientist called Hans Christian Gram, and he basically wanted to distinguish the different types of bacteria, and that is how he had developed this staining technique. And then through this staining technique, He is able to distinguish bacteria into two main classes. They were the gram positive and the gram negative, and the ones which were not staining well with the gram stain, they were put under a third category, which was the acid fast stain, and which under which category come our very famous tuberculosis or TB bacilli. Okay, so next we go on to the differences between the gram positive and the gram negative bacteria. So what are the gram positive bacteria appear as? What is their appearance? They are distinctive in their appearance by having a purple color after staining, whereas gram negative bacteria, they have a pale reddish color after staining. And what is the reason for the staining? Because they are most importantly having a thicker cell wall and a thicker outer layer, the gram positive bacteria, whereas the gram negative bacteria, they are having a much thinner layer and they are uh, able to have the stain. They are not able to retain the stain properly. So the reason for why we are dividing these bacteria into gram positive and gram negative is that the gram positive has a thicker cell wall and the gram negative have a thinner cell wall. Okay, so examples of gram positive bacteria: all the staphylococci, streptococci, pneumococci, and the listeria species, they come under gram positive bacteria. So it's not a very big class, it's a small class, but compared to gram negative bacteria, which is a much bigger class, and in this come most of the bad ones like the E. coli, Vibrio, Klebsiella, Pseudomonas, Salmonella, all of these species come under the gram negative bacteria. So what is basically the difference between the cell walls of these? Let us just go into that. The gram positive bacteria, they have no outer lipid membrane, whereas in gram negative bacteria, the lipid membrane is present. And the gram positive bacteria, they don't have any O specific side chains present, whereas the gram negative bacteria are having O specific side chains present. And teichoic and lipoteichoic acids are present in gram positive bacteria, which are not present in gram negative bacteria. So basically, as I said again, gram positive bacteria have a thicker cell wall, gram negative bacteria have a thinner cell wall, because of which they are unable to retain the stain and then they appear as pink, whereas gram positive bacteria are able to retain the stain because of a thicker cell wall and they appear purple. Okay, so going on to the next branch, virology, very famous, especially in the last few years, taking much more of a notorious role because of the COVID pandemic. So these are actually different from other microbial groups. They are all different. They are different from every other microbial group, like the bacteria, fungi, parasites. They are different from all of them. Basically, the reason is that they are firstly non-cellular. They are not cellular organisms, and then they are so small that they are filterable. That means that they can filter through all of the bacterial filters that have been discovered. And thirdly, because of their very small size, they can be seen only with an electron microscope. So they are not visible through the regular microscope that is used to see the bacteria. So they can only be seen through an electron microscope. And viruses, because they are acellular or not cellular, they can only reproduce by using the cellular machinery of their host. So whoever they infect, then that person or that animal cells are used for the reproduction of the viruses. So this is the reason why they are also called obligate intracellular parasites. So these are some of the features of viruses. First of all, they are particles composed of an internal core. So that's why we say that they are not cells, but they are particles. They have an internal core, and this internal core contain one of the two nucleic acids. They either contain the DNA or they contain the RNA. And based on this, viruses have been classified into these two major classes, the DNA viruses and the RNA viruses. Example of these DNA viruses are the herpes viruses, and example of RNA viruses is our very notorious COVID virus and also the HIV virus. All of these are in the category of RNA viruses. So viruses do not, of course, have a regular, as we said, regular structures like the cytoplasm, mitochondria, ribosomes, and nucleus. They are just particles which are multiplying inside the host organism. Okay, then viruses, as we said, they have to reproduce within cells because they cannot generate energy or synthesize proteins of their own. So they can, because of this, they are called obligate intracellular parasites. Then viruses replicate in a manner different from that of other cells. So they do not go into binary fission where only from one virus two viruses are formed. So instead of that, what happens is each virus can divide into many hundreds of progeny viruses, and that is the reason why the multiplication of viruses is much faster, and the disease spreads also much faster compared to bacterial diseases. Coming to the next branch, which is immunology. So immunology is study of how the body protects itself against infectious diseases, because if suppose the body is being attacked by organisms like it, be them bacteria, be them viruses, be them fungi, then there has to be some mechanism which is inbuilt in the body from nature to fight that infection or to resist that infection. So this mechanism is basically called the immune system, and the study of this is called immunology. And important initial barriers to infection are now, so we are going to just talk about what are the basic structures through which the body is firstly fighting. So there are firstly structures that fight an infection, and then there are cells which fight infection in the body. So these are all of them, they are composed of the immune system. Okay, so what are the physical structures? Firstly, the skin itself is a physical structure which stops any of these organisms from getting into the body. And then our various body secretions such as saliva and tears, which we uh, associate mostly with emotions, but they actually have are an important part of our immune system because they contain molecules that can neutralize bacteria. Okay, so and then as we just said, the other mucosal tissues like lungs and airways and even the intestines are lined with mucus that are able to trap potential infectants. And in the airways, mobile ciliate hair will be present, which can actually trap this bacteria and virus and the fungi and prevent them from reaching the essential organs. Okay, so and then tissues such as the skin, mucosal surfaces, and airways also contain populations of immune cells. So this is the second part of the immune system, which are the immune cells that can again respond to infectants that breach these physical defenses. So if suppose these infectants are able to breach these physical defenses and actually reach the cells, then at that time these immune cells come into picture and they uh, carry out the fight against these microorganisms. And then, what are the components of the immune system? It consists of two branches. First is the innate immune system, which utilizes certain hardwired strategies. That means that these are the pre existing system in the body, as we discussed some of the physical barriers that we discussed, skin, mucous membrane, then the secretions. These are all part of the innate immune system, which is actually existing, pre existing there. And whenever they are alerted by certain typical signals of infection, they come into action and they cause, then they carry out the protection of the body. And then second is the adaptive immune system. Now, what is the adaptive immune system? This is able to develop highly specific responses. So these responses are developed in response to an infection. So they are not existing, they are not pre-existing. They are developed in response to a invasion or to an attack by a microorganism. So what happens is when these microorganisms attack, then that when the infection reaches a target, then there is an extraordinary response to this infection, and then the body produces some specific cells, and these are part of the adaptive immune system, and it relies mainly on the innate immune system to alert it. So the innate system alerts it, and then the adaptive immune system carries out the attack on these organisms, and then shapes its response according to them. Then next branch is mycology. Mycology is the study of fungi, and they include several thousand species of, as we said, they are eukaryotic and they are spore bearing organisms. They obtain their simple organic compounds by absorption. They have no chlorophyll, so they are different from plants because they don't have any chlorophyll, and they reproduce by both sexual and asexual means. Okay, so fungi, why are they? Why is the study of fungi called mycology? Just a small tidbit that Mycos means mushroom in Greek, and that is the reason why they have named it not as fungology as would be expected of it. It is called mycology. Okay, and what is the specific features of fungi? They are usually filamentous, and their cell walls have chitin. This is what main. This is the main ingredient that keeps them separate from other organisms. So their cell walls are having chitin, and study of fungi is called mycology, and the fungal diseases are also called as mycosis. Okay, so together with bacteria, they are the major decomposers of organic material in the soil. So fungi also play a very important role in deposition of organic matters in the soil. So if suppose somebody is burying a dead body in the soil, then decomposition of that dead body is carried out by bacteria and to a great extent by fungi also. So they degrade these complex organic matter into simple organic and inorganic compounds. And in doing so, they help recycle carbon, nitrogen, phosphorus, and other elements to be reused by other organisms. So this is a nature cycle of nature that nature carries out to maintain a balance. Okay, so they also cause many plant diseases. So they are a big danger to agriculture also. So they cause agricultural diseases also, and also of course several human diseases, especially of the skin. We have diseases like dermatophytic infections which are caused by fungi. And as far as classification goes, fungi are made up of two major groups. They are the molds and the yeasts. Molds are basically filamentous fungi, and yeasts are small unicellular fungi. They are not multicellular fungi. So molds are the multicellular fungi and the filamentous ones, whereas the yeasts are the unicellular fungi. Okay, so two major groups of organisms make up the fungi. As we said, they are the molds and the yeasts. So the molds are the multicellular, as we said, they consist of long branching filaments of cells, and these long branches are called hyphae. And when a tangled bunch of hyphae comes together, this is called a mycelium. So the hyphae and the mycelium are the ones which consist of these molds. Okay, so these fungi are said to be sinusitic fungi, and those fungi that have cross walls are called septate fungi. So if you see a fungus, and then if you see a diagram of a fungus and there are septate walls on that, that means they are known as septate fungi. Okay, so next are the yeast. Second definition of this, the division of fungi are the yeasts. And yeasts are microscopic, and as we said in just now, we said unicellular fungi with a single nucleus and eukaryotic organelles. So they reproduce asexually by the process of budding. So simple budding, very simple reproduction process in the yeast. Just by budding, they reproduce, and examples of these yeasts are the uh, very so the examples are the candida and the mushrooms that are also consumed. So candida basically cause the disease, whereas there are fungi which are useful also, such as I just said, the mushrooms. So this is as far as the yeast and the fungi are concerned. And finally, let us go to the study of parasites, which is called parasitology. This includes the study of three major groups of animals. They are the protozoa, the helminths, which are the worms, and then the arthropods that directly cause disease, or they also act as vectors of various pathogens. Okay, so a parasite, as we all may be knowing, it is a pathogen that simultaneously injures and also derives sustenance from its host. So, compared to a saprophyte, a parasite does not cause any advantage to the host. It only causes disease and it only causes loss to the host. So that is why it is called a parasite. So it lives on the host, it derives its sustenance from the host, and then causes disease to the host. So this is what a parasite is. And then some organisms called parasites are actually commensals. Commensals is they neither benefit nor harm their host. Example of these are entamoeba coli. So two words that need to be remembered in this context are the parasites and the commensals. And infections of humans that are caused by parasites, there are many. There are millions of infections that are caused by parasites, and they range in severity from being relatively innocuous, not having any symptoms, to some which are actually fatal. So, the diseases caused by these parasites constitute major human health problems throughout the world. Of course, we know about the seriousness of infection of malaria. It's a very serious problem, especially in African countries. And some worms like the Ascaris lumbricoides, also known as the roundworm, that is also a cause of disease, in especially in children. And then the incidence of many parasitic diseases, example malaria and schistosomiasis, have increased rather than decreased in recent years, and there have been efforts to find vaccines to this. Especially a malarial vaccine has now been discovered, and it is going to become global in a short period of time, probably. And then other parasitic illnesses have increased in importance as a result of the AIDS epidemic, because whenever the body's immunity is decreased, as it happens in AIDS, the body becomes more susceptible to infections by parasites, especially the parasite known as the Pneumocystis carinii, which causes pneumonia, and strongyloidiasis, which is a worm which causes diarrhea. So these are the different types of parasites that cause disease in immunocompromised individuals. And of course, the migration which is happening very commonly nowadays compared to the previous times, people moving from one country to another, this is one of the another reason because of the spread of these parasitic infections from areas of high prevalence to areas of low prevalence. And this is why these diseases now are getting more global, and they are not limited to only certain countries anymore. Okay, so that was a small introduction to microbiology. I hope it gives you an idea of what the subject is, and I hope that in subsequent presentations, we'll go into much more detail of each of these branches of microbiology. And hope you had a good time, and I will see you again. Thank you.